Greetings. Today we're going to talk about the development of our sexual anatomy in utero, in the uterus, as before we are born. To give you a sense of where we're at now, we just finished talking about sex and genetic diversity, and we're moving into the biological aspects of our sexuality. Before I get started, I need to make sure that we're clear on some very important terminology. Now, sex is our biological role in reproduction. It's also an action, and we're going to talk about that later in the semester. But in terms of identifying our biological sex, this is what we're talking about. Which role do we play in reproduction to bring on the next generation? This should never be confused with our gender or our gender identity. That is our sense, our internal conviction of either being female or male or neither or both. All of those are real, and they are not necessarily connected to our biological sex. There's also sexual orientation. That's referring to who we are attracted to, who we form relationships with or fall in love with. And then there's also gender roles. Gender roles are a societal concept because every society assigns different roles to people depending on what their gender is. We're going to talk about all of these things later on in the course. Now, there are connections between our biological sex and our gender. So our biological sex, whether we are born male or female, this influences our gender identity, but it does not determine it. What that means is that our biological or our assigned sex is typically aligned with our gender. They're typically the same. And we call this cisgender. But it's not always the case. And when someone's biological sex and their gender differ, we say that they are transgender. There are also biological or connections between biological sex and our orientation. Our biological sex influences who we will be attracted to or love, but it does not determine it. What that means is that people are more typically attracted to the other sex, which we call heterosexuality, but not always. There are people who are attracted to the same sex, homosexuality, to both sexes, bisexuality, to neither, asexuality, and all kinds of other sexualities, orientations that we're going to talk about later in the course. So, we'll get to gender and orientation later. First, we need to understand how we identify biological sex. So, identifying sex at birth, which you may sometimes see abbreviated as SAB, we are assigned either male or female at birth based on the appearance of our genitals. Okay, here is a typical human female, typical human male. These pictures are very hard to come by, so I apologize for the lack of diversity. However, our genitals don't always align with our internal anatomy. For instance, some individuals have male genitalia while they have internal female structures, and the other way around. And some individuals are born with indeterminate or ambiguous genitalia. Now, I've put both of these terms in quotes because if you are the owner of said genitalia, it probably doesn't feel very good for them to be referred to as ambiguous or indeterminate. They're your genitals, right? So why they need to have a label at all, I'm not sure. But this is where, and we will talk about this as I go on, but this is where the shame starts in terms of people who fall outside of the typical genital structure. Okay, well, so genitals maybe aren't the best way to do this. Let's see if we can think of something else. How about chromosomes, right? Got the X chromosome and the Y chromosome. And it's true that while generally a person with two X chromosomes will be female anatomically, and one with an X and Y chromosome will be male, it is also the case that some individuals will have XY chromosomes and female anatomy, and some individuals will be born with two X chromosomes and have male anatomy. So our chromosomes aren't going to do the job either. All right, well, how about let's just use another way to determine a baby's sex? Well, we can't use sperm or egg production because they're not doing that yet. Plus, that would be pretty creepy. And investigating their internal anatomy, whether they have ovaries or testes, a uterus or not, isn't practical or really desirable. It is possible to see some of these things on an ultrasound, but it's not extremely accurate, and we're sure not going to open up every baby's belly just to see what's inside before we send them home with their parents. All right, so how do biologists determine sex if everything's kind of wishy-washy? Biologists define sex based on whether an individual has motile or movable gametes or immotile, ones that can't move themselves. So motile gametes are sperm and immotile are eggs. So to a biologist, if you're making eggs, you're female. If you're making sperm, you're male. All right, well, why don't biologists just base sex determination based on the genital appearance? Because in some species, the females have penises, so the genitals don't always predict our reproductive role. Female elephants often have a penile structure, and so it may not be possible to identify the sex of the elephant just based on what you see hanging between their legs. Female hyenas, and this is a female, there's the penis, have penises that they use for mounting and penetration just like the males do. It's just there's no sperm there. And there are insects where the female has the penis and the male has something that resembles a vagina. She inserts the penis and extracts the sperm. And she either uses it to make babies or she eats it because, there's not a lot of resources where they live. So we can't use genitals. At least not across the board. The point is that society's way of determining sex isn't scientifically accurate. Just looking at the genitals, we know, is not an accurate way to predict someone's sex. And biological sex isn't a straightforward thing anyway. It's not either or. There are lots of options. And this begs the question, why are we determining sex anyway? Really, think about it. Is our biological sex really important? And if so, what for and when? Do I need to know whether a baby is a boy or a girl in order to buy the right diapers, to find the right formula, to find the right clothes? Well, you could argue that, but that's a gender role thing. Really, when and where do we need to know someone's sex? And it's really only around reproduction. All right, so setting aside the question of whether we should determine someone's sex, which we will talk about more as we go on, let's try to at least wrap our heads around how our biological sex develops. Okay, so our biological sex actually comprises three things. Our genetic sex, I'm covering the words again, which means that we're looking at the chromosomes and the genes that we have. Our anatomical sex, which means we're looking at the genitals and the internal anatomy. And our physiological sex, what gonads we have, whether we have testes or ovaries, and whether we're producing sperm or eggs. All right, so that's a lot of things to consider when you identify someone's biological sex. Much more complex than just looking at the genitals. Okay, so it all starts with our chromosomes, which we talked a bit about last time. Now, we're going to ignore all of these other 22 volumes of chromosomes and just focus for the moment on our sex chromosomes. we have two sex chromosomes. We have an X chromosome, which is rather big, and we have a Y chromosome, which is rather small. When an individual inherits two X chromosomes, they generally develop as female. And when an individual inherits an X and a Y, they generally develop as male. So here we see, here's an egg-producing cell that produces two eggs. See, each one got one copy of that chromosome. Here we have, of the X chromosome. And here we have a sperm-producing cell that has an X and a Y. So half the sperm are going to end up with the X chromosome. Half the sperm will end up with a Y chromosome. Now, if this egg fuses with this sperm, right, so both of those are coming here, then we have an individual with two X chromosomes, so most likely female. If this egg and this sperm fuse together, same thing. But if either of these eggs fuse together with the sperm that has the Y chromosome, then we have an XY individual who is most likely to develop as male. So, the simple version of the story is that two X chromosomes will give us a female offspring, and an X and a Y will give us male. It's worth mentioning that it's only the father that has the Y chromosome that determines the sex of the offspring. The mother, or person with two X chromosomes, can only give an X. The father has an X and a Y, and therefore it is the father that determines the sex of the offspring. I find this interesting because King Henry VIII is well known for divorcing, beheading, or just imprisoning his wives, many of them, because they couldn't produce a male heir. But it was really on him. They just didn't know that at the time, or he didn't care. Okay, so we know that our genetic sex is usually determined by the presence or absence of the Y chromosome. So the Y chromosome has a switch in it that allows us to go from female development to male development. Let's take a look at how that works. All right, a tiny little thing does all this. Okay, so here we have an X chromosome and a Y chromosome. Now, there's a gene on the Y chromosome that's called SRY, or sex-determining region of the Y chromosome. When I was in college, I used to remember this by saying, "Sorry, you're having a boy," which kind of told you what kind of phase I was in. I don't really feel that way. Anyway, we have the SRY gene, and the SRY gene makes a protein that causes the gonads. All right, the gonads are primitive structures that can either become ovaries or testes. It causes the gonads to choose the path of becoming testes. And these testes then begin to secrete, God, there's nowhere to put my screen here. Okay, then begin to secrete two things. One is called Mullerian inhibiting factor, or MIF, and the other is called testosterone. And these two things are what direct, the path of development for the internal and the external anatomy. So it all starts with the SRY gene. Without this gene, the gonads automatically differentiate into ovaries. That's their default path. Okay, so if the SRY gene isn't there, or it's there and it doesn't do its job, then the gonads are automatically programmed to become ovaries. Now, our internal and our external sexual anatomy develop separately and independently, which is why they don't always match. Now, I'm about to go through some slides that talk about where these structures come from. So I want to, since we haven't done the adult anatomy yet, just give you a sense of what I mean by internal and external anatomy. All right, let's start with the male. So in the male, the testes are where the sperm are produced. After the sperm are produced, they're stored in a structure that's wrapped around the testis called the epididymis. You'll hear that. And the sperm move from the epididymis to these tubes called the vas deferens. The vas deferens loop around the bladder, join up with a few glands that produce secretions that end up in the semen, and then they come out through a tube called the urethra. So that the sperm and the semen can exit the body. Now, the urethra is the same tube that carries urine from the bladder. So in males, there is, and we'll talk about this later, basically a switch that prevents the two from mixing, ideally. Now, external anatomy for the male really is referring to the penis and the scrotum with the testes. The internal anatomy is talking about the vas deferens and these glands. And the epididymis. Forgot the epididymis. Now, female anatomy. The external female anatomy is known as the vulva. Okay? You may have heard people describe this as the vagina, but the vagina is actually internal. The vagina is a tube. What's on the outside is a vulva, and it includes labia, or lips, 2 pairs, an outer pair and an inner pair, and a clitoris, and a fold of skin that covers the clitoris, known as the prepuce, or the hood. That's the external. The internal female anatomy is the vagina, the cervix, which is the opening to the uterus, the fallopian tubes, also known as uterine tubes, and then, of course, the ovaries, where the egg are produced. Okay, so in both male and female development, the internal anatomy is controlled separately and independently from the external anatomy. All right. At 6 weeks of development as embryos, this is what our internal reproductive anatomy looks like. All of us have gonads, which are bipotential. They have the potential to become ovaries and the potential to become testes. We don't know which way they're going to go yet. There are also 2 structures, one male and one female. The female structures, which you see here in red, are called the Mullerian ducts. And the male structures, which you see here in blue, are called the Wolffian ducts. We can just call them male and female, but I do want you to see this word Mullerian because it's also in the name of one of the chemicals, hormones that's secreted in development. Now, male or female, both of these tubes connect with this structure, which is called the cloaca. Cloaca is a term for an opening that serves 2 purposes, reproductive and excretory, meaning getting rid of the urine. The cloaca eventually becomes the bladder and the urethra, the tube that carries the urine to the outside of the body. And in females, the cloaca also forms the outer third of the vagina. So when I said external and internal anatomy are controlled separately and independently, even the vagina has 2 parts. The inner 2/3 goes with internal anatomy, and the outer third develops with the external anatomy. You know, because we like to make it complicated. Okay, so if our embryo inherits the SRY gene, it causes these gonads, that can be either thing, to develop into testes. And the testes begin to produce Mullerian inhibiting factor. Now, just the name tells you what it's going to do. Mullerian inhibiting factor is going to cause the Mullerian, or the female ducts, to disintegrate. All right, so now only the male is around. The testes also produce testosterone, and testosterone helps to protect the Wolffian, or the male ducts, so that they don't disintegrate. Okay? Now, the male ducts, the Wolffian ducts, eventually develop into the epididymis and the vas deferens. The epididymis is where the sperm mature and are stored until ejaculation, and the vas deferens are the tubes that carry the sperm from the epididymis to the urethra, which is going to be how they get out of the body. The urethra also transports urine. So in males, there is one shared opening for the urinary system and the reproductive system. Now, if the embryo does not inherit the SRY gene, the gonads become ovaries. Ovaries secrete estrogen, but not testosterone. And the lack of testosterone means that the male ducts disintegrate. They need testosterone to protect them and keep them from being destroyed. And also, as there's no MIF, or Mullerian inhibiting factor, the Mullerian ducts are not under attack, and they're going to stick around. So, the female ducts then develop into the uterus, the upper part of the vagina, the cervix, and the fallopian or uterine tubes. The vagina, which you can see here, connects directly to the outside of the body. It doesn't join together with the urethra like we saw in males. The urethra also, I don't know where to put this picture, the urethra also connects directly to the outside of the body. And so in females, there are two separate openings for urinary and reproductive purposes. All right. I want you to think about this, though we're going to talk about it when we meet in class. But based on what I've described so far, just about the internal anatomy, I want you to see if you can predict what would happen if we have a person with XY chromosomes that produces testosterone but doesn't produce Mullerian inhibiting factor. And then see if you can figure out what would happen if we have an XY person that produces Mullerian inhibiting factor and testosterone, but their cells don't respond to testosterone, which is kind of like not having testosterone at all. Or what happens when an XY person inherits a mutated, non-functioning copy of the SRY gene? All of these things happen. Okay. Let's shift gears a little bit and look at the external sexual anatomy. Where in the internal sexual anatomy, we had both male and female parts. All of us had both, and then one survives and one disappears. In the external anatomy, we all have the same set of structures at the beginning, and they change one way in the presence of testosterone or high levels of testosterone, and they change another way in the presence of estrogen. So all of our genitalia start off looking like this. There are no male and female parts. These are rudimentary tissues that have the ability to develop either way. Now, before I move on to talk about how they develop, I just want to help you orient yourself to this picture. So these are the infant's or the embryo's legs going off in either direction. And what you're seeing right here is a cross-section or a cut-through of the vertebrae. Early in development, our spines extend beyond our body. We basically have tails. And as we develop and grow, the spine compresses into that triangle-shaped bone that's at the base of your spine called the sacrum. But what we're seeing here is the tail that we all have. And by the way, when we talk about two structures, two adult or mature structures that come from the same tissue, we say that they are homologs. All right, homo means same. So they're coming from the same tissue. Okay, so when the embryo has more testosterone than estrogen, these structures develop and move to the left side of the screen. When an embryo has more estrogen, the structures develop more towards the right of the screen. So don't worry about the names of any of these. This yellow structure here, in the presence of testosterone, eventually becomes what's known as the glans, or the head of the penis. Has lots of nerve endings, very sensitive. In the presence of more estrogen, that yellow structure again becomes what's called the glans, also known as the clitoris. And it too has lots of nerve endings and is very sensitive. They have similar properties because they came from the same tissue at the beginning. All right, let's look at these red structures. So in the presence of testosterone, these red structures, you can see here that they're sort of forming a tube and wrapping around. So the red structure in the presence of testosterone becomes the urethra that runs through the shaft of the penis. And what you can't see here, but you can kind of see here, is that it also forms the tissue surrounding the anus. It's just our artist has cut it off down here. Same thing in females. This red tissue makes a tube, starts to fold, but then it unwraps and becomes the labia minora, or the inner lips of the vulva. Okay, so the only difference really is that here it stayed a tube and is internal, and here that tube opened in order to create the labia. Okay, these turquoise structures. In the presence of testosterone, these become the skin that covers the shaft of the penis, including the foreskin, which covers the head of the penis, and in an erect penis is pulled back to expose the glands. In the presence of estrogen, that becomes the skin, like the foreskin on the penis, but the skin that covers the clitoris, also known as the hood. Both of these have the same scientific name, which is prepuce, which I think is one of the ugliest words you could come up with to describe sexual anatomy. So, anyway, let's take a look at this purple tissue. It develops a little bit later, so we don't see it in our 8-week embryo. But in the presence of testosterone, this purple tissue develops into the scrotum that surrounds the testes. In the presence of estrogen, it becomes the labia majora, or the lips that are on the outside of the vulva. Again, because these two tissues come from the same original source, they have very similar properties in terms of their sensitivity and appearance. Okay, so let's talk a little bit more about these hormones that are making all of this happen. Now, we know that testes produce testosterone, ovaries produce estrogen, but those two hormones are not just produced in the gonads. We also have these glands called adrenal glands. Ad means on top of, renal is kidney. So these are the things that look like little hats on top of our kidneys, and they produce a number of things, but among them are estrogen and testosterone. Not a lot, but some. So, typical female having ovaries would produce lots of estrogen, but also a little bit of testosterone. Typical male having testes would produce lots of testosterone, but also a little estrogen. Now, the other thing you need to know is that in adults, the level of testosterone varies tremendously from person to person. Now, women have a range of testosterone secretions coming from the adrenal glands that is a fairly small range. Men have a much wider range of testosterone secretion or testosterone production. The point is that the difference between women and men in terms of testosterone is smaller than the difference between a man that produces lower levels of testosterone and one that produces higher levels. So there's so much variation in testosterone secretion. That's your take-home message. Now, what does testosterone do? Well, testosterone is produced in the testes, and then it's released and it goes into the bloodstream, which carries it to target cells around the body. Inside the target cells, there are receptors. When testosterone binds its receptor, both of them move to a part of the DNA that contains a gene that we want to make, that testosterone is stimulating the production of. So we end up with a particular protein or proteins based on the fact that testosterone has arrived. Now, there are a lot of things that can affect testosterone activity. You can produce some testosterone, a little bit from the adrenal glands, a lot from the testes. You can produce more or even more. There's a lot of variation there. Now, testosterone doesn't function as it is first formed. It actually has to be activated. And so there's going to be a difference from one individual to another in terms of how this activation takes place. For some people, they can produce testosterone, but it never gets converted to its active form, or most of it doesn't get converted to its active form. And then we can have differences in terms of the receptors. Some people have receptors that are less sensitive to testosterone or less obedient when testosterone arrives than other folks. Sometimes these receptors aren't responsive at all. So you may as well not make testosterone because nobody's going to listen to you. Okay? All of that affects the degree to which the cells produce the proteins that give us the traits that we associate with testosterone. All right. So, an individual can have a whole bunch of testosterone and a little bit of estrogen, and their genitals are probably going to resemble what you see at the top of the screen. Or an individual can have a small amount of testosterone, maybe a lot of estrogen, and their genitals will appear similar to what you see, I don't know where to move this again, on the bottom of the screen. But the thing is, because of all the ways that testosterone can vary from one individual to the next, there's all kinds of concentrations of testosterone that we can have. And what that means is that because we all produce testosterone, and because the concentrations vary widely between individuals, and because our genitals all come from the same tissues, genitals that appear between female and male are natural variations. They are not only possible, but expected. Again, I don't know where to put that camera. Okay, so our most typical forms are here, where we have low testosterone, high estrogen, and here where we have high testosterone, low estrogen. But because there are so many ways for the levels to vary, all of the in-between structures are expected. Okay, so now think about this with the external anatomy, just based on what I told you about how it works. How would it be different if an XY person didn't produce testosterone or didn't have functional receptors? How is that going to change the way their genitals appear? Or what would happen if you have a person with two X chromosomes, but their adrenal glands produce way more testosterone than normal? How are their genitals going to look different? Or an XY person, where their adrenal glands produce way more estrogen than what is typical. All right, so puzzle on that for a little bit. Okay, so all of this was really complicated and has introduced more questions than answers, probably. So it leaves us with, how do we determine a person's biological sex? Now, using what we call the binary model, which is what's most popular in our society and many societies around the globe, it's very simple. It's either or. You have two X chromosomes, you're female, an X and a Y, you're male. If you're female, your gonads are ovaries. If you're male, they're testes. Your hormones, very clear, estrogen for the ladies and testosterone for the men. Your internal organs, If you're female, you have a uterus. If you're male, you don't. Genitalia, if you're female, you have a vulva. Male, you have a penis and scrotum. And society even goes so far as to say our gender identity is always totally aligned with that. That if you have all of these, then you see yourself as a girl or a woman. And if you have all of these, then you see yourself as a boy or a man. So, if we had little sliders for measuring things to help us figure out biological sex, the binary model says that all of the sliders are going to be at this end, you're female, or this end, you're male. But the reality is that there are many places where these things are not clearly on one side or the other, and therefore it's not clear how we would identify a person's biological sex if we only have this binary model. Okay. Now, all of these differences are part of the diversity that our biology works to generate. I'm going to repeat this over and over again. There is no right way to be male or female. There is no need to be either male or female. The differences are evidence that our species is doing what it needs to survive. And if we see these differences as expected rather than as mistakes, it allows us to be more accepting of ourselves and of others.